Hello friends, Vanakkam. Welcome to Whiteboard Learning with me. Today we discuss the topic neurotransmission in human brain. Neurotransmission means நரம்பிய கடத்தல். அதாவது ஒரு சிக்னல் ஒரு நரம்புல இருந்து இன்னொரு நரம்புக்கு எப்படி கடத்தப்படுது. ஸோ நிறைய நியூரான் சேர்ந்து தான் ஒரு நரம்பை உருவாகுது. ஸோ ஒவ்வொரு நரம்பை தாண்டி தான் அந்த சிக்னல் போய் நம்ம பிரெயினை சேருது. அதை பற்றி படிக்கலாம். நியூரோ டிரான்ஸ்மிஷன். ஸோ பிரெயின் ஆக்டிவிட்டி இஸ் மேட் பாசிபிள் பை த இன்டர்கனெக்ஷன்ஸ் ஆஃப் neurons that are linked together to reach their targets. A neuron consists of cell body, axon and dendrites. See, this is the structure of neuron. So this is dendrites, this is the cell body, this is axon, this is axon terminal. So this is soma, nucleus, axon, node of Renvier, Schwann cell, myelin sheath. So this is axon terminal. So types of neurons are sensory neuron, interneuron and motor neuron. So the structure of three types of neurons. See, this is dendrites, direction of conduction. This is cell body, this is axon, this is cell body, this is axon, this is cell body, dendrites, this is axon. That means, see, sensory nerves, function of sensory nerves, it passes the impulse senses to brain. Motor nerves, it passes impulse brain to all muscles. Mixed nerves, it function of both sensory and motor nerves. So this is the sensory neuron, this is interneuron, this is motor neuron. So brain to cell body to brain, cell body to cell body, brain to cell body. Sensory neurons, it passes impulse cell body to cell body, interneuron, and it passes impulse cell body to brain is motor neurons. Then dendrites are often extensive branches that receive information in the form of signals from the axon terminal of their neurons. The signals received may cause the neurons to initiate an action potential. That means an electrochemical signals are nerve impulses. Action potential means an electrochemical signals or nerve impulses. We see elaborately about nerve impulses. So which is sent along its axon to axon terminal to connect with the dendrites or with the cell body of another neuron. Then an action potential is initiated at the initial segments of an axon which contains a specialized complex of proteins. When an action potential reaches the axon terminal, it triggers the release of neurotransmitter at a synapse that propagates a signal that acts on the target cell. See, this is the neuron. See, this is one neuron, this is another neuron. This first neuron or presynaptic neuron. The second neuron is postsynaptic neuron. That means, see, this is the portion of the impulse between one neuron to another neuron. See, this is called synapse. This is axon terminal. This is neurotransmitter after release. See, this is the structure of neuron. Already we know this is dendrites, cell body, nucleus, axon, hillock, axon, direction of signals, axon terminals. This is the synapse, dendrites. This is postsynaptic neuron, axon, collateral axon, axon terminals. Then action potential. How to action potential in pre and post synaptic neurons. First, action potential arrives at axon terminals. So, action potential arrives at axon terminal. Second, same, this is calcium channel, Ca2 plus channel. So, voltage gated Ca2 plus channel open. Then, third, this is Ca2 plus enters the presynaptic neuron. It enters the presynaptic neuron. After Ca2 plus signals to neurotransmitter vesicle. It signals to neurotransmitter vesicle. This is synaptic vesicles or neurotransmitter vesicles. So these are the neurotransmitters. Then after fifth, vesicles move to the membrane and dock. So move to the membrane and dock. Then sixth one, see, neurotransmitters release via exocytosis. Neurotransmitter release via exocytosis. So it released the process of release named exocytosis. So it release in between pre and postsynaptic neurons. Then seventh, neurotransmitters bind to receptor. It is bind to receptor. So these are the receptors. First receptor, second receptor, third receptor, etc. and so on. This is the docking protein. So supportive protein. Finally, signals initiated in postsynaptic cells. Then the signals initiated into postsynaptic cells. So then next, see the another diagram. This is dendrite axon. This is synapse and dendrite axon. Synapse. These are the action potential. Axon and dendrites. So presynaptic and postsynaptic neurons. So these are the formation of neurotransmitter vesicle. This is neurotransmitter. These are the vesicle. Then Vesicle fused with membrane exocytosis. These are the exocytosis process. It releases the space in between space of axon and dendrites. These are the neurotransmitters. These are the calcium channel. So see, then neurotransmitters released into synapse. Then fourth one, neurotransmitter diffuse across synapse and bind with receptor on dendrites. So this is the process in between axon and dendrites. See, these chemical neurotransmitters include dopamine, serotonin, GABA. GABA means GABA uses and side effects. See, GABA means gamma aminobutyric acid. Is a chemical that is made in the brain and also found in some foods. In the brain, GABA has anti-seizure and anti-anxiety effects. So this is important. Then see, these chemical neurotransmitters include dopamine, serotonin, GABA, glutamate, and acetylcholine. GABA is the major inhibitory neurotransmitter in the brain, and glutamate is the major excitatory neurotransmitter. Neurons link at synapse to form neural pathway. Neural circuits and large elaborate network systems such as salience network and the default mode network, and the activity between them is driven by the process of neurotransmission. Then GABA works by blocking brain signals, neurotransmission. So GABA works by blocking brain signals, neurotransmission. There is interest in use GABA as a dietary supplement, but when taken by mouth, GABA may not be able to cross the blood-brain barrier. Therefore, it is unknown if GABA supplements have effects on the brain. People use GABA for many different conditions include stress, anxiety, depression, high blood pressure, but there is no good scientific evidence to support its use. Then we see some diagrams of action potential. See presynaptic neuron, postsynaptic neuron. So in this diagram is at rest phase. This is active phase. So in rest phase, it is no works. See, synaptic vesicle, neurotransmitter. This is peptide molecule, synaptic cleft, plasma membrane, enzymes, receptor, Ca2 plus channel, voltage gated. So this is a receptor, this is enzyme. Then this is the active presynaptic neurons. So this is the action potential. So second, Ca2 plus channel open, and third neurotransmitter into synaptic vesicle. Then this is main important one. See, already we study exocytosis. See, this is the function of exocytosis phase. Then fifth, see, receptor binds with neurotransmitter. Then sixth, some enzymes bind with neurotransmitter and some released into in between space. So seventh, neurotransmitter go into the action into the presynaptic neurons. So this is presynaptic neuron and postsynaptic neuron. Then elaborately we study the action potential. See, early endosome into, see, H plus ATP is converted by ADP, the presence of enzymes. Then it's into reserve pool. This is neurotransmitters. This is SOT. This is VAMP. So main functions: docking, priming, hemifusion, intermediate, fusion, fission, and final neurotransmission binds to postsynaptic cells. See, this is MUNC18. This is syntaxin, SNAP25 are the active zone. Then see, these are the Ca2 plus channels. Then these are the complexin. Then docking phase. Then next phase is priming phase. See the position of SYT and VAMP. See, then it's converted into other function, hemifusion intermediate. So it's near end of the presynaptic neuron. Then fusion release, neurotransmitter release, and it goes to the channel into postsynaptic neuron. So these are the some neurotransmitters go into the fission. So this is clamping, this is uncoating, this vesicle go to ready for another impulse. This is direct recycling into H plus ATP into ADP. So these are the action potential phase, neurotransmission in brain, and action potential. Thank you.